Welcome back. So now that we've taken a look at the membrane itself and looked at the protein channels that are involved in moving the ions in order to make up our membrane potential, we can take a closer look at the actual membrane potential itself. So the main membrane potential is essentially a process of combining a chemical gradient with a electrical gradient. And so we have this process here where we see in terms of the chemicals, we have areas where we have less concentration and more concentration of two different colored particles here. And so the blue ones being more concentrated down in this area, the red ones being more concentrated up in this area. And so we have this concentration gradient. The red ones, in a sense, want to move in this direction, and the blue ones want to move in this direction. So that we get a process where we're changing that concentration, and eventually they're going to be equal on either side. And the same thing goes for an electrical gradient as well. We've got more negative ions over here, and they want to move towards the positive. And the same thing then goes for the more negative ones. They want to move towards the more positive, or the more positive ones want to move more towards the more negative ones. And this is all allowing us, if we have nothing in a sense as a barrier, this is going to allow that to happen. So they're going to move towards that lesser concentration and balance everything out. And so eventually they're going to come and have a balanced charge on either side here. And so we have the same number of positives, we have the same number of negative structures that are there on either side of our membrane. And we can change that and alter what we have as far as the particles are. And we can take 5 of those positives and move them to the other side. And now we have a relative difference between those two spaces. And so this side is more positive, and this side is more negative relative to each other, because we have a difference there in the amount of particles that are there. And so if we were then to take, and rather than have a true barrier there where things can't move across there, and put a membrane in between the two, this membrane then allows us to potentially move substances across that membrane itself that's there. We're going to see that where we have that difference. And so once again, over here we have the excess amounts of positives. Over here we have more negatives than we have positives. And so we have a situation where on either side of that membrane, the opposites are going to become attracted to one another. And so we're going to have positives and negatives on either side. And we're going to have this relative charge that we can even measure between the two sides as well. The rest of the space inside those two chambers, in a sense, or inside the two cells, is going to be basically neutral. And so all of the positives and negatives in these spaces here are going to then take and separate themselves out as evenly as possible, so that we have the same amount of charge everywhere, and it's basically completely neutral all throughout. The only place where we're going to see that charge in the two is in between the membrane itself, where they can line themselves up together. And so this is essentially what we get with cells. And so if we have this circular cell here, we have the inside of the cell, we have the outside of the cell, and depending upon which one has more or less of those ions and the charges that are there, we're going to have the charges line up just along the very edge. So we're going to have positive and negatives along those edges there. In our cells, we've talked about some different ions before in terms of things that have charge. And so we've talked about things like our sodium, potassium, chloride ions, all having charge to them. And so these are going to be found in different concentrations in different places. We've talked about the sodium potassium pump before, and so we looked at that and how the sodium moves 3 sodium out and 2 potassium into the cells as that pump is running. And so this is going to create the situation where for the sodium, we have a higher concentration outside of the cell in the interstitial fluid. And for the potassium, we have a higher concentration in the cytosol, so inside the cell itself. we have a higher concentration of that potassium that's there. Chloride ions are also going to be involved in this makeup of the charges as well. And chloride ions are going to be out in the outer portion of the cell as well, and so it's going to be more in the interstitial fluid that is there. And for our other negative ion, this is actually protein. And so protein does have a charge to it, and those proteins are in much greater quantity in the cytosol. And so depending upon the makeup of each one of those is going to dictate whether we have a positive or a negative within the cells themselves. So when it comes down to it, why are we making these membrane potentials in the first place? Who needs them? For the most part, it's muscles and nerves. And so both of them are excitable tissues. They're excitable when it comes to a potential energy from electrical components. And so they have the ability to propagate an action potential along the surface of those cells. And so we can get this process where electricity will actually flow across the cells and down through the cells and to wherever it needs to go. In nerves, this is done in terms of what we call action potentials. And so we have this action potential that's going to travel from the cell body all the way down the axon to its axon terminals. And that electrical charge that's moving along the length there then eventually gets to the end, and we send, in a sense, a message. We send some sort of information. In muscle, that action potential is what's going to cause muscles to contract. And so it's that electrical impulse that's going to essentially turn on muscle contraction. And so in those muscles, this is what's going to allow for the muscle to eventually shorten. So we talked a little bit about the ions, and primarily what we're going to focus on in terms of those ions are sodium and potassium. And so, as I said, we have a greater number of sodium ions on the outside and a smaller number of potassium ions on the outside. And then on the inside, we have greater amounts of potassium and relatively small amounts of sodium. So the sodium is going to want to get into the inside of the cell, and the potassium is going to want to get out of the cell. And we talked before about leak channels, and in terms of those leak channels, the sodium has fewer leak channels, so it comes back in slowly, and actually the potassium leaks out relatively quickly. So it has a much greater ability to leak out than that of the sodium there. And so even though we're putting 3 sodium out, 2 potassium in, with that potassium leaking out much faster, it starts to come on the outside as well. So we're getting more and more positive ions on the outside of our cell. They're starting to accumulate and build up on the outside there compared to the inside. So this creates this resting membrane potential. And so we end up having a positive charge on the outside of the cell and a negative charge on the inside of the cell. And so what this amounts to a certain degree is we can kind of start to think about it as simply being the numbers of ions. And so we say that a resting membrane potential in neurons is at roughly negative 70 millivolts. And so one way to kind of think about this is just to think about it, because sodium is going to be the big thing that moves, is we can think when we compare the inside of the cell to the outside of the cell in terms of sodium, we can think about it as far as, say, the outside has 70 more than the inside. So we kind of have 70 on the outside and none on the inside, or something like that. Or however many we have, if we even just make it 100 for each, then there's 170 on the outside. There's more of the positive ions on the outside than it is on the inside. So when we compare the two there, we're going to have an excess amount of those sodium ions on the outside. So we've got more of those positive ions out here from the sodium, and the sodium is trying to leak back in through a leak channel, but it's a relatively slow process. The potassium leaks out. It's a relatively quick process for that. And again, we have excess amounts of negative proteins on the inside of the cell compared to those chloride ions on the outside. So the inside becomes a negative situation. We have a negative side to it. And so we've seen things that have positive and negative on either side of a membrane before. And so we have a battery. And effectively, this is kind of what we get here, is that we have a battery in these cells. They're constantly recharged by the sodium-potassium pump, which does require energy. So we do put some energy into it, so we're recharging that cell. And as we move those ions, it's similar to if we connected a wire to the two sides of the battery there, and maybe put a bulb in between there, and then that light bulb is then going to shine. In this case, we're going to generate an action potential with our energy. And so we're going to have that electrical potential travel along the length of the cell and eventually open up some channels elsewhere on the cell itself. So we can change the membrane potential by altering the channels. And so we can change the channels themselves. We can open some up. We can close some, and we can start to change that voltage. So we started out with negative 70 millivolts. And if we start to allow some of that sodium to come into the inside of the cell, we can start to get what we call depolarization. And so depolarization is going to cause a change in that cell to where now we're allowing those positive ions to come in, and the voltage is becoming less negative. And so down here on our voltage, we have negative 70. Make this one negative 65. And we come back down and then go back up. So this is what we get, is what we call a graded potential. So the change itself is not enough to get us an actual action potential. And so the voltage change, in order to get there, we would have to have negative 60 millivolts. And then the voltage would then go up from there. So if we got a change in negative 60 millivolts, we'd then have our action potential. But we don't get that here because we have not opened up sufficient amounts of ion channels. So we only have a slight depolarization of the cell. At the same time, we can also generate hyperpolarizations. And so again, if we are starting at negative 70 millivolts, and we cause that voltage to go down even further, we now have a hyperpolarization. And so hyperpolarization is where we make the membrane more negative on the inside of the cell. And so we have more negative ions that are coming into the inside of the cell. So we have something like gated chloride ions coming through a gated channel, moving into the inside of the cell. And so they're coming into the inside of the cell and making the inside more and more negative. This is going to make it less likely that we're going to get an action potential. And so the cell itself is more negative in terms of getting up to that negative 60 millivolts at our, we can make that green for go. So in order to get up to that negative 60 millivolts, normally we just have to have a change of 10. Now we have to have a change of 20 millivolts. And so we're going to have to move, in a sense, 20 more ions of sodium into the inside of the cell in order to get that depolarization to occur, in order to get the depolarization to the point of threshold, so we can have an action potential. So the actual action potential itself, we've got to get to a point where we reach this threshold. Threshold has to be gotten to. And so with that, if we have graded potentials, and so once again, if we're at negative 70 millivolts, negative 65 millivolts, negative 60 millivolts, and we're starting out here at negative 70, we come up to 65, maybe 64, and we're going along like this. These are all graded potentials. But once we get up to negative 60 millivolts, then we've reached threshold, and the process itself is going to go up all the way to a positive 30. So we're going to make our way up to a positive 30. And once that happens, we can't stop it. It's kind of like tipping over dominoes. We can't stop this process. It's all or none at this point. And so once we get it, we get it. We're going to have that action potential. And for our neuron, that action potential is going to travel all the way down the axon, all the way to its axon hillox, all the way to the very end of the axon itself, to the terminal points of that axon that's there. And once we start it, we can't stop it. Just like with dominoes, once you start it, you can't stop it. Unless there's something broken, in a sense, in that neuron, then we may stop that action potential. But as long as everything is working the way that it's supposed to, that action potential is going to go, and it's going to go all the way until the end of the cell. This process begins at the axon hillock. And so the axon hillock here, this initial segment of our axon, is the place where the graded potentials are coming in here. And so we're getting up to our, eventually up to our positive 60 millivolts. And once we get up to that positive 60 millivolts, then we're going to move along the conductive segment. And so we're going to travel along this conductive segment. We've reached the tipping point, and now it's going to travel all the way down to the axon terminals here. And we're going to get to the transmissive segment here. And this is where this one neuron is going to pass, in a sense, its information onto the next cell. And so it's going to tell the next cell to do something, typically by releasing neurotransmitter. So it's going to release a neurochemical onto the next cell, and that's going to tell that next cell to happen. All the rest of this neuron here is all part of its receptive segment. And so this is where we're going to have different connections to other cells. Those other cells are going to make their connection there, and they're going to all create graded potentials. And so graded potentials are going to occur. If those graded potentials summate, and we get summation of those graded potentials are generally going to be less than negative 60 millivolts. But as soon as it equals negative 60 millivolts, then that's going to take us to our action potential. And so that's going to summate up to that action potential, and we can allow for that signal to be then transmitted. So threshold begins when we have sodium gates opening up. And so those sodium gates are starting to open up. And this is our activation gate here. That activation gate is going to open up, and that's going to cause all of the sodium ions to start to rush to the inside of the cell. So sodium is going to start to move into the cell. Then our potassium gates are going to open. So this gate is going to open here. And potassium, which is in excess amounts on the inside of the cell, is going to flow out of the cell. So it's going to start to move out. And that's going to take us from our negative 60 millivolts all the way up to a positive 30 millivolts at that point. So we're going to make a very large change in the voltage once we've reached that threshold. So we're going to get up to that threshold of negative 60 millivolts, and then that's going to send us into an action potential as soon as we get to that point there. So negative 60 millivolts, our activation gate opens up. So our activation gate here is open. We also have an inactivation gate. That one is open at this point as well. So the sodium is able to come through. At rest, that inactivation gate is open also. So it stays open all the way up until we hit positive 30 millivolts. At positive 30 millivolts, that inactivation gate is going to close, and that's going to bring us to, in a sense, stop the influx of sodium. The potassium channels are going to stay open, and so those potassium channels are going to open at this point, and they're going to allow for potassium to exit the cell. So we created a scenario where the inside of the cell at this point is positive. We've got to get rid of some of that positive ion and move them out by opening up those potassium channels. Once we hyperpolarize the cell, we get down to negative 90 millivolts, our inactivation gate is going to reopen, the activation gate is going to close, and now the cell is hyperpolarized. In order for this cell to have another action potential occur, now we've got to go from negative 90 all the way up to negative 60. So now we've got to have a change in 30 millivolts in order for that to happen. So it's going to take a very strong stimulus in order to get that to occur. Our sodium potassium pump is now working, and our sodium potassium pump is pumping our sodium and our potassium back, and we get back to our resting potential. We're now back to where we started at. And so everything is reset. Now it only takes, again, that 10 millivolt change, and our cell can then start to have another action potential at that point. And this happens every single time that that action potential occurs. Every time a nerve, in a sense, sends a signal, this whole process happens. And so it's happening very, very, very fast. And so we're talking about milliseconds in time, just a few milliseconds for all of those processes to occur. So during the action potential itself, potassium is going to start to leave out of the cell. This is going to alter the cell itself, and voltage-gated sodium channels are going to be open at this point, and that's going to trigger more and more of these channels to become opened. And so as we have a change in voltage, those voltage-gated channels start to open up. And so this is what's going to help to propagate our action potential down the axon. And so as we have our cell there, this is what's going to happen to move it down. So all along that axon, we have our voltage-gated sodium channels. And so those voltage-gated sodium channels are sensitive to the fact that we're going from a negative to a positive. We have our negative all the way down, and the next one turns on, next one turns on, and then eventually we start to repolarize along the way. And our action potential keeps on moving its way down until it reaches the very end there. And so the process itself is occurring all the way along its length. We have an area where we are going through depolarization. And then right behind that depolarization, we're starting to do repolarization. And so we're going to come back around to a process where that repolarization is occurring, and we're getting back into a negative state. And that's going to eventually create all along the length there as it propagates its way down the cell itself. And so this is what ensures part of that all or nothing process. And so once we have an action potential beginning, it's going to travel all the way down, because these are, in a sense, like our dominoes. And so as we change the cell itself, and we start to make that cell go to positive in this area, as we make that change there, then the next one becomes positive, and the next one becomes positive, and the next one, and the next one, next one, next one, next one, and right behind it, we then make it all negative. And so we bring it right back into repolarization, and the inside of the cell is repolarized. This is where we're actually doing the depolarization, and it's heading on its way down. It can't go back on the axon. It can only go in a single direction. It can only go from that axon hillock towards the axon terminals. And that's going to ensure that one-way direction of our signal, sending it all the way down towards the end there. So getting back to that rest point that we mentioned there, we have our sodium potassium pump. That sodium potassium pump is running. It's pumping our 3 sodiums out. It's pumping our 2 potassiums in. And this is going to take us from that negative 90 millivolts and bring us back up to negative 70 millivolts. This is going to take a little bit of time. So we're going to have this refractory period where it's going to take either, when we have a relative refractory period, so we can have a relative refractory period, and this is where in that relative refractory period, this is where we can go from that negative 90 up to negative 60 and still get a action potential. But we also have an absolute refractory period. And in this one, no amount of stimulation is going to get us a action potential. This is going to require, in terms of that absolute refractory period, we have to reset the sodium channel in order to get into the relative refractory period. So that sodium channel, not our leak channels, but the gated channel that we had that looks something like this, that has to reset in order for us to be able to once again get into this relative refractory period. So the absolute refractory period is the period of time from when we started our action potential all the way up until we have resetting of the sodium channels. So the activation gates need to be reset, and then the relative refractory period can occur. So we have two different portions for that refractory period itself. Absolute, no matter how hard we stimulate that cell, nothing's going to happen. Relative refractory period, a sufficiently strong action stimulus can get us to the action potential once again. But again, that's where we've got to go from that negative 90 all the way up to negative 60 in order to get there. So we have to have a change in the strength of the stimulus in order for us to do that. Normally, we're going to go from that negative 70 all the way up to a positive 30. And that's going to be, in a sense, a 100 millivolt change. Now, if we're all the way down at negative 90, now we've got to do a 120 millivolt change. So it takes a much stronger stimulus in order for us to get there. So better to be in that relative refractory period than need to have another action potential to come along within the cell itself.